Hi, my name is Catherine Métayer. I am professor of epidemiology at the University of California, Berkeley. The goal of my research is to better understand how the environment and diet can influence the risk of cancer in children.
My name is Joseph Wiemels, and I'm a professor at the Center for Genetic Epidemiology at the University of Southern California. My research examines the developmental and genetic causes of childhood leukemia, brain cancer, and sarcomas.
Cancers in children between 0 and 19 years of age are heterogeneous, affecting various organs. The most common type is leukemia, representing about 25% of all cancers, followed by brain tumors, also called central nervous system tumors, CNS. About 5,600 new cases of leukemia and brain tumors are diagnosed every year in the United States. Rarer types include cancer of the liver, kidney, retina, bone, and soft tissues, which are also grouped under non-CNS embryonal tumors. The age distribution of childhood cancers varies by type. Early onset cancers that develop mostly before age 6 include leukemia, brain tumors, and embryonal tumors, lending support to the prenatal origins of these cancers, as well as the impact of early life events. Other cancers occur at a later age, mimicking cancers seen in older patients, such as testicular germ cell tumors and epithelial cancer of the thyroid. Childhood leukemia includes acute lymphoblastic leukemia, called ALL, the most common type, followed by acute myeloid leukemia, called AML. The age of onset distribution of these cancers varies largely, with a peak incidence of ALL between age 2 to 5, while the rates of AML are more or less constant, suggesting different timing in cancer initiation and progression. Prenatal origin of childhood ALL has been the most studied to date.
A focus on the prenatal period for the initiation of pre-leukemia was first demonstrated in twin studies. Here, we showed that two monozygotic twin girls who contracted leukemia at age 5 and 13, respectively, shared the same exact genetic mutation called a translocation, showing that the mutation occurred before they were born when they shared a common blood circulation in the womb. This twin pair demonstrated that leukemia can originate during fetal gestation, but then that it requires additional genetic mutations that may occur many years after birth. The timing of these mutations is also instructive. The natural history of leukemia causation includes both prenatal and postnatal genetic and epigenetic changes, contributing to the two-hit hypothesis for ALL, meaning two distinct windows of vulnerability for these hits. We now focus on the prenatal period as especially sensitive for these leukemia-initiating events or mutational errors that lead to pre-leukemia cells. After birth, these cells may progress to overt leukemia disease once additional mutations occur, which may result from environmental exposures or damaging infections during childhood. Childhood leukemia, like most human cancers, has a diverse array of genetic mutational subtypes, which share the leukemia blast cell phenotype, but differ in prognosis and age at onset. Here, we show the most frequent subtypes by age group along the top, and note that the vast majority of infant and childhood subtypes have already been shown to have a prenatal origin, which is also probably true for some adult forms of the disease as well. Modern therapies are now tailored to these specific subtypes, leading to increasingly positive outcomes for leukemia patients. These subtypes are also extremely valuable in our attempts to link specific disease subtypes to epidemiologic risk factors.
National data on cancer incidence rates show that children of different racial and ethnic backgrounds develop different types of cancer. For example, Latinx children in bright blue in this graph are at high risk of having leukemia and germ cell tumors of the testis, whereas Black children in brown color in this graph have the lowest rates of those cancer types. In contrast, white children in dark blue in the graph are more likely to have lymphomas and brain tumors. Rates of cancers for other children, Asians and Pacific Islanders in pink and American Indians in green, also largely vary by cancer type. Trends in cancer incidence over time differ by race and ethnic group. While the rates were going up for all children until 2016, they seem to go down for whites and to some extent Black children. In contrast, Hispanic, Asian, and Pacific Islander children still experience increasing rates of cancer overall. This graph shows the types of cancer that are increasing in children, namely leukemia, non-Hodgkin's lymphoma, some non-malignant brain tumors, such as pilocytic astrocytomas, germ cell tumors like testicular cancer, and epithelial tumors like thyroid cancer. Regarding childhood leukemia, a significant upward trend is seen among Latinx children only. There is some indication that a similar trend is observed in Black children and Asian Pacific Islanders, although non-significant. In contrast, incidence rate of leukemia is decreasing in white children. The occurrence of childhood cancers also varies by region, as illustrated by this graph, using various colors to represent various cancer sites. For example, Black children in sub-Saharan Africa and in the United States have the lowest rates of leukemia in dark blue. Children in Europe and Northern America have the highest rates of brain tumors in gray, while Native American and Southeastern Asian children have low rates of lymphoma in brown. We also see geographical patterns in the United States, where rates of childhood cancers vary by state, as shown by the gradient of blue, from high rates in dark blue to low rates in white. Descriptive statistics like I presented are important to generate hypotheses to better understand why occurrence of childhood cancers change over time and by region. It is most likely a combination of lifestyle, environmental and social factors, as well as genetics. Indeed, for example, rates of obesity in the US are dramatically high in certain populations and states, especially in Latinx, as shown in the left graph. These rates parallel increasing rates of chronic diseases such as cancer. The Latinx population is also experiencing a high burden of pollution and social deprivation, as shown in this graph using the CalEnviroscreen mapping tool that collects environmental, health, and socioeconomic information that identify California communities that are most affected. An overall CES score was created to integrate various measures, and in this case, we see that the higher CES index on the vertical axis is observed in areas mostly inhabited by Latinx on the horizontal axis. We conducted a childhood leukemia study in California and collected dust samples in the homes of Latinx and non-Latinx white families to measure over 50 persistent chemicals such as pesticides and flame retardants found in furniture. The squares above the red line indicate that more chemicals were detected in Latinx homes compared to non-Latinx whites, supporting again the observation that Latinx communities have disproportionate exposures to potentially harmful chemicals.
The incidence rates of several childhood cancers have increased in recent decades in the U.S., and the burden varies by racial and ethnic group, suggesting the important contribution of the environment at large, alone or in combination with genetic susceptibility. Early life exposures from the time of preconception and fetal development to early years of life are likely to contribute to the initiation and/or progression of cancer later in life. These findings suggest opportunities for public health and behavioral interventions to reduce the burden of cancer in children and families.